have an actual title for my talk other than Basic Science 1. So thanks for the invitation to be here and to contribute. Certainly two very impressive talks before me, one on the clinical side and then, of course, that personal perspective from Scherer. So want to make sure that, I welcome you actually to home turf here in Dallas. This is where we are. And I want to make my fat cell stance for you as well. And obviously, we are very focused on the fat cell and try to sort of link some of the things we've heard towards the molecular physiology of the adipocyte. Now, given the massive macroscopic changes that are associated with lipedema, you would think it should be relatively trivial for the molecular physiologist or the cell biologist to figure out what is going on at the level of adipose tissue. And there are things that hint at the fact that it might be more difficult given that in 2023 we still lack a clinical marker, a diagnostic with a plasma marker that we can measure or a biopsy that we can take and subject to a test. We don't have that, and hence the whole confusion, of course, between what is lipedema and what is just obesity. Now, from the patient level, it seems like, given what we've heard, that if you know what you're looking at with the appropriate background, that you can actually diagnose visually a patient, and certainly with a few additional office assessments, you can reach that conclusion. But it has been incredibly challenging to find out more about the underlying pathology. And I want to just touch upon 3-4 small vignettes without showing you any data, basically, to sort of highlight what is going on here. So when we look at a cohort here, and again, this is mostly based on clinical samples that were collected by Karen Herbst, and we've been very fortunate actually to be associated with the Lipedema Foundation pretty much from the beginning in terms of having access to samples and being able to contribute. You can see, of course, the overall progression with the BMI, with the different stages that we've heard. This is frequently seen, but not a must. You can also see the distribution pre- and post-menopausal for the different stages on the right-hand side. So that's actually an important distinction as well, as we'll see the hormonal changes may, at least in part, play a role there as well. And the two molecules that we are dealing with as fat cell-derived markers that are easily assessed in circulation, adiponectin and leptin, are shown here. And you can see that there are basic rules for these two. So leptin levels in circulation are proportional to fat mass in most cases, not always, but very frequently. You can see on the right-hand side that this kind of, when you plot the BMI versus leptin levels across the different stages, that this pretty much holds true. Adiponectin is actually exactly the opposite. That's what it's famous for. It's an adipocyte, a fat tissue-derived secretory marker. But the more fat tissue we have, the less adiponectin is in circulation. So frequently people actually use the ratio of leptin to adiponectin to actually come up with a digitized view of the whole thing. But what you can see here, particularly in the stage 1 samples, is that the basic rules don't really seem to apply to adiponectin. And it's somewhat reminiscent of the population that several groups have defined, but Dr. Klein has been really on the leading edge of defining the metabolically healthy obese phenotype in a classical setting where things become fairly independent of fat mass. It actually can be protective in many ways, and you have somebody with an excessively high BMI that has absolutely no manifestations of metabolic dysfunction. And that, again, based on what we've heard from the clinical picture and the personal assessment, a lipedema patient is quite frequently a metabolically healthy, obese individual, which doesn't help the cause in terms of finding the underlying pathology, because we can certainly look at dysfunctional obesity and lean functional fat tissue and reach certain conclusions. And when we now, for instance, eliminate the postmenopausal subjects here, it doesn't help. Leptin still correlates quite well. Adiponectin doesn't seem to follow the conventional rules. So the underlying reasons for that are, of course, not quite clear. Now, transcriptomic analysis of lipedema fat tissue has been undertaken in a number of different cohorts. Papers have been published, and a lot of thought has been put into that. And at this point, we are very much used to looking at omics data, whether that's transcriptomics, proteomics, metabolomics for that matter. And we've done that in a number of different cohorts now, and I am embarrassed to admit that there's just nothing that strikes you as particularly unusual. And that is a problem. I mean, do you see transcriptional changes when you go through these biopsies and do RNA-seq on them? Absolutely. You can come up with pathways. There are certain specific hits that will go up, others go down. Do you see a general pattern? No. Does it hold up across different cohorts, different samples collected by different clinics? Usually no. There might be certain trends, but there's certainly nothing that would excite me at this point to say, aha, this is probably the reason. Do we have dysregulation clearly at the level of the extracellular matrix? We do see these trends. It is an ECM disease, but what causes it, what drives it, we don't know. Is there perhaps a hint of a pro-inflammatory phenotype in that adipose tissue? Absolutely as well, but again, it doesn't strike you in any particular way. So I want to spare you any long lists of potential hits out there. There's nothing that we could at this point really firmly point at. Proteomics has been done, and again, this was done in collaboration with Karen Herbst's samples as well, trying to sort of go in and find out what's going on. In terms of principal component analysis, yes, based on the proteomics data, we can actually see differences between lipedema patients and controls. There are some interesting trends in there, but again, no home run in that context. Perhaps one thing I want to point out, and I realize you can't read that, but those are all the components of the complement fixation cascade, which are generally down-regulated in the context of lipedema. And I'll come back to that because there might be actually some significance to that in this context as well. And last but not least, for someone like me, we like to have a model system. We do work with mice. We manipulate the genetics of mice, and we've learned an awful lot about the pathophysiology of obesity from mouse tissues. We can very much mimic the metabolically healthy, obese phenotype that Dr. Klein describes in the clinic. We have equivalent models in the preclinical setting. We have, of course, the metabolically unhealthy obese mouse. We have the metabolically unhealthy lean mouse. So we can do all these things. But as far as a lipedema mouse model is concerned, that's a far stretch at this point as well. In fact, even if we had a lipedema mouse model, how would we know we actually have a lipedema mouse model? Because we cannot translate the clinical marker into the preclinical setting. So we have a lot of different ways by which we can induce things in an adult mouse, and we can learn and try to learn from the transcriptomics and metabolomics and more importantly proteomics from results from that have been published and that we see in our own data. There seems to be a dysregulation, again, referring to the vasculature. VEGF is a critical component, a pro-angiogenic component. An excess of VEGF, a disproportional excess of VEGF can actually have negative consequences by increasing vascular permeability. We've also done a lot of work on hyaluronic acid synthase. Hyaluronic acid is a very, very hygroscopic molecule that we have in our pericellular environment, binding water. and that can have both positive and negative effects basically on the local tissue. And with these doxycycline-inducible models, we can create mice that actually express one or the other or both of these and manifest some reflection of a lipidemic phenotype. But again, we've been analyzing these mice at every level transcriptionally as well, and we are just not sure yet whether that's a good preclinical model for the clinical state. And that's a question that comes up, of course, with any preclinical model, whether that's a fatty liver disease, whether that's a cardiac phenotype, a kidney phenotype. What is the appropriate mouse model to use? And because the clinical signature is still so ill-defined for lipedema, it is probably reaching too high at this point to even attempt to make a decent preclinical model to reflect that. Now, again, we looked at a lot of histology across the board. This is now from obese controls, abdominal fat samples. Characteristic on the left hand, this is an H&E. On the right-hand side, you see a stain, a trichrome stain that highlights the extracellular matrix a little bit more. This is what we usually see, and this would be distinct from a lean individual. But when we move over to a stage 3, the latest stage sample here from abdominal tissue, there's really nothing we walk away with. It looks exactly like a normal fat tissue, and if we knew what to stain for, we would probably done that, but we've done a lot of immunohistochemistry and really haven't come across an awful lot of distinct, unique features. Next level up is electron microscopy. You look at the fat tissue there at very high magnification. You see a wonderful set here, for instance, of these caveolae. We have control obese versus stage 3 lipedema. Every now and then you see a structure and say, oh my God, yes, this is clearly distinct. But then you look across a lot of different samples, you do a lot of EM, and there's really nothing that sticks in terms of a unique morphology of the fat tissue or the interstitial tissue in between. The best we can do here is the lipedema Pac-Man. This is a unique structure, or more literally a Pac-woman, basically, which happens to be, but there is nothing that translates across a larger set of pictures. So even the EM leaves us quite puzzled. So in the absence of actual tangible data, you can just go to the desk, go away from the bench and start thinking and postulate and try to connect dots in terms of what it is that could be happening out there. And then once you have a model, you can then go back and basically perhaps do some of the tests. And we've done that actually quite a few times. You can see here we published thought pieces that we believe are reasonably well-founded on some facts out there, but most of this still needs to be actually tested experimentally. But the key proteins that we keep stumbling into when we try to connect these dots involve a protein called caveolin, which is very, very abundant in the normal adipocyte, and then a matrix metalloprotease, MMP14. And there is some interesting biology there that lends itself to be dysregulated in the context of lipedema. Caveolin interacts with the estrogen receptor. There is a population of the estrogen receptor ER-alpha that's bound to the plasma membrane. Caveolin mediates that binding. It has to be released for the estrogen receptor to go to the nucleus and actually do its thing. And then, on the other hand, the exoplasmic MMP14 is also interacting with caveolin 1, and the release of MMP14 into the interstitial space has actually all kinds of consequences as well that would fit the overall appearance of what's going on in lipedema. So there's an interesting set there to start out with. And very recently, this is a paper that just came out in Nature Reviews Endocrinology about a week ago or two. We are proposing a new, an added-on sort of theory where we invoke the complement fixation cascade as part of the pathophysiology. And that's not restricted to just lipedema. we would argue that it's been known for the past 25, 30 years that adipose tissue and the adipocyte very specifically, the most abundant components that the adipocytes expressed are components of the complement fixation cascade. C3, complement factor D. As you know, this is an immune component that is supposed to basically become activated and then integrate into the pathogen membrane, and it forms at the end of the day what we call a MAC, which is a membrane attack complex, which will lyse the pathogen. Now, this is not just for a pathogen, this actually goes for mammalian cells as well. And of course, the vast abundance of the complement components in the local microenvironment has never been addressed and has never been actually explained why we would have that. And we postulate here that this will be an integral part of sustaining the cell number of adipocytes basically within adipose tissue, tightly regulated, that there is a small but regular turnover of fat cells, and the activation of the MAC, this attack complex, is central to the maintenance of adipose tissue cellularity. And there are a number of different ways by which you can control the activity of this complex, including, for instance, that downregulation of the complement cascade members, including factor D, for instance, and that goes back to the proteomics data that I've shown you. A characteristic feature of lipedema is the downregulation of the complement fixation cascade, which is quite unique. And then you have a number of other ways by which you can control the activity of this complex through interaction with inhibitory molecules, internalization again through the caveolin mediated pathway in caveolae, as well as other functions there. So the additional component that I want to add here is lipopolysaccharide. So this is bacterial debris, LPS, basically, and as you will see in a second, the fat tissue in adipose tissue, the perigluteal fat tissue has actually a lot of receptors for lipopolysaccharide. And one of the ideas that we would like to put out there, again, it's consistent with a lot of the clinical phenomenology that you see out there, is that during certain changes, whether that's a pregnancy, menopause, or other hormonal changes, that the leakiness of the gut is profoundly affected. We know that, for instance, during pregnancy, endotoxin levels in circulation go up, and they will reach higher levels in circulation, including in fat, and that we would postulate, given based on a bunch of additional data, that this leads to an expansion of adipose tissue and actually affects this macrophage, this membrane attack complex in a negative way as well, leading to hyperproliferation, hyperplasia, and hypertrophy of some of these structures in adipose tissue. So the adipocyte per se overexpresses the receptor for LPS, that's TLR4, and we and others have published on this, and you can knock the TLR4 out from the adipocyte, and it has profound metabolic consequences. So the sensing of lipopolysaccharide within the adipocyte seems to be an integral part of the physiology of adipose tissue and makes major contribution. And again, these LPS levels in circulation fluctuate depending on the specific physiological state of the patient. So during a pregnancy, again, we have higher levels of LPS. The same during menopause. A leaky gut leads to enhanced exposure of this gluteal fat, and this is a particularly abundant source for TLR4. So not every fat cell expresses the same level of TLR4. That perigluteal region actually is particularly prone to have high levels of TLR4, and that's why it would hyper-respond basically to that LPS. And then in combination with this local overabundance of the complement fixation cascade lead to specific, site-specific changes in that particular regions within the leg. So the challenge for us is to try to explain the local differences with regards to the different fat paths. Why don't we have a symmetrical response across all fat paths? And why does it only hit the very specific regions that it hits? Because, for instance, the rule of thumb is for weight loss in the average individual, weight loss is always symmetrical across all of the fat paths. So you can't target the weight loss procedure specifically towards visceral or towards subcutaneous fat. But here we have a disease which is not symmetrical across all fat paths. And hence, this has to be blamed in part due to the differential gene expression that you see in different regions, including now TLR4 in this particular context. And again, without going further into the thought process there, that this will ultimately affect the turnover of fat cells, the argument being that in the case of lipedema, we have senescent old fat cells hanging around. They don't turn over, they proliferate, so to speak, in terms of increased recruitment from precursors, and hence the disproportionate expansion. So this is a combination of LPS, varying at different stages of the life cycle, LPS, together with the hormonal impact of estrogen, caveolin, MMP14. We believe those are the players that we want to pay attention to going forward in terms of actually establishing a more functional connection between all of those. And at the end of the day, again, it does involve an inflammatory component here. The inflammasome is part of it, and again, caveolin interactions there play a role as well. So in other cell types, this has been fairly well worked out, but in the context of the dysfunctional adipose tissue, we aren't quite there yet in terms of figuring out what exactly is going on. And then PPR gamma, the key adipogenic transcription factor, is actually being induced by this inflammasome as well as a repressor. We have enhanced PPR gamma activity. And again, that's in principle a good thing, but not in the context of this one-way street towards hyperproliferation of the fat tissue in this particular context. So I beg for forgiveness for being overly speculative, but this is not by choice, but this is simply based on the fact that we really lack a good direction, a good molecular direction in terms of what's going on within the pathophysiology of lipidemic adipose tissue, and we need new ideas out there that can be put to the test in terms of candidates, because it is extremely challenging. Lipedema is not the only fat tissue disease that behaves like that. Even the trivial aspects like cellulite, even we don't understand cellulite. We don't know what to do about cellulite. Do we need to do something about cellulite? Well, it's not a disease. So there is not really that level of priority. But in the context of lipedema, given the manifestations that we've heard about clinically, and given the burden on the patient, obviously, this is something that we believe really deserves a lot of attention going in the future. It's a huge challenge to really pinpoint what it is. Is it a single gene? Is it multiple genes? Are we going to come up with a biomarker in plasma or in a biopsy that we can pinpoint as a unique and reliable marker for lipedema? That is still something in the future. With that, I want to thank my collaborators. This has been driven on our side by Leon Straub, Noel, and Gilfer mostly. I would want to thank Karen for her samples over the year, and my colleague Sam Klein, who's always an infinite source of wisdom and samples. Or is it just samples? I don't know about the wisdom part. But and then my colleague Ilja Kruglikov, who is the mind behind many of these more theoretical connections that I pointed out. Thank you.